Cells constantly send and receive messages to communicate with each other. Typically, one cell produces chemical substances as messengers, called ligands, and another cell, called the target cell, receives them using a receptor. Receptors are specialized proteins located on the cell membrane or inside the cell. They have a specific binding site shaped precisely to fit the ligand. This specificity ensures that communication is precise and cells respond appropriately to the signals received. Notice the non-target cell does not have receptors and therefore cannot respond to the ligand. Here we can see a receptor. This receptor will only bind ligands that match the shape, charge, and chemical properties of its binding site, such as this one. Once the ligand binds to the receptor, it activates the receptor. This can lead to a change in shape or trigger a response inside the cell. This response is the start of what's called a signal transduction pathway. Hopefully, as you are thinking about ligands and receptors, you are drawing comparisons to other molecules you already know about, such as enzymes and substrates. Let's compare and contrast enzymes to receptors. Chemically, both molecules are proteins. Similar to the enzymes, receptors also have a specific site where the ligand can bind. In addition, the shape and chemical properties of the ligand binding site match those of the ligand, just like an enzyme and its substrate. Finally, both enzymes and receptors are unchanged by the binding of a ligand, even if there are temporary changes to induce fit. Unlike enzymes, which catalyze chemical reactions and change their substrates, receptors typically do not alter the ligand. Communication is essential in living organisms. Even single-celled prokaryotes such as bacteria communicate with each other. One way bacteria do this is through a process called quorum sensing. Quorum sensing is a form of cell-to-cell communication that bacteria use to detect their population density and coordinate behavior as a group. It's a powerful example of how simple cells can act collectively. Each individual bacterium releases small signaling molecules called autoinducers into its environment. At low population densities, these molecules simply diffuse away, and their concentration remains too low to trigger a response. But as the number of bacteria increases, the concentration of autoinducers also increases and begins to build up. When it reaches a threshold level, these molecules bind to receptors in or on the bacteria cells, activating a signal transduction pathway. This pathway leads to the activation of specific genes, but again, this only happens when the population is large enough. The behavior that results is something the group couldn't achieve as individuals. Individually, a bacterium doesn't gain much from producing certain products like toxins or enzymes. Doing so alone would waste energy. But when all the bacteria coordinate their efforts using quorum sensing, they can act like a multicellular organism, behaving as a group. This shift from individual to group behavior allows them to take on complex tasks like forming biofilms, attacking a host, or emitting light. Let's take a closer look at bioluminescence in the marine bacteria Vibrio fischeri. This was the first case of quorum sensing to be discovered. Vibrio fischeri cells produce and secrete an autoinducer which can bind to a receptor protein called LuxR. When this binding occurs, it turns on the Lux operon. This causes the transcription of genes, which result in the production of the enzyme luciferase. Luciferase catalyzes an oxidation reaction that releases energy, and more than 80% of this energy is transformed to a greenish-blue light. Free-living Vibrio fischeri maintain a fairly small population density, so they do not receive enough of the autoinducer to produce luciferase. Light is therefore not emitted, as it would be a waste of energy. However, Vibrio fischeri can also be found living inside the Hawaiian bobtail squid. They form a mutualistic relationship with this animal host, meaning both organisms benefit. In the squid, the bacteria live inside a structure called the light organ. During the day, the squid hides in the sand, but at night it comes out to hunt. Moonlight can cast a shadow from the squid and alerts its predators. Vibrio fischeri, however, can help produce light that matches the moonlight above. This provides the squid with a form of camouflage and allows it to hunt more effectively and avoid its own predators in the process. And the bacteria benefit from the safe, nutrient-rich environment provided by the squid. This coordinated response benefits both species. In animals, communication between cells is essential for everything from growth and metabolism to muscle movement and immune defense. To accomplish this, the body uses a variety of signaling chemicals. Since these chemicals vary so much chemically, they are usually classified according to their function rather than their structure. Let's look at 4 major categories of functions. First, hormones are signaling molecules secreted by endocrine glands into the bloodstream. They travel long distances and reach target cells throughout the body. Hormones are released by specialized organs called glands. For example, the adrenal glands secrete epinephrine or adrenaline, which travels through the blood to organs like the heart and lungs and triggers the fight or flight response. Next, we have neurotransmitters, which are chemical messengers released by neurons to send signals across synaptic gaps between nerve cells or from nerve cells to muscles or glands. Neurotransmitters are released by presynaptic neurons into the synapse. They bind to receptors on the postsynaptic cell. The gap between two neurons at a synapse is between 20 and 40 nanometers, and most neurotransmitters only travel this short distance. This happens in a fraction of a second, so neurotransmitters convey their signal far more quickly than hormones. They are also rapidly broken down in the synaptic gap, or reabsorbed by the presynaptic neuron, so they only persist for a fraction of a second. Therefore, their effects are short-lived. Acetylcholine is an example of a neurotransmitter you will learn more about in the video C2.2 neural signaling. Cytokines are signaling proteins secreted by a wide range of cells. The same cytokine may be secreted by different cell types, and one cell type may secrete many different cytokines. Cytokines act on nearby cells or even on the same cell that secreted them, and therefore do not travel as far as hormones. They are large proteins and cannot enter the cell, so they bind to receptors in the plasma membrane of a target cell. This binding causes a cascade of signaling inside the target cells, which leads to changes in gene expression and ultimately cell activity. Cytokines play a key role in inflammation and other responses in the immune system. They also control cell growth and development. Calcium ions are unique because they often function inside cells as second messengers, triggering rapid responses in both muscle fibers and neurons. In muscle fibers, calcium ions play a key role in muscle contraction, which you'll learn about in more detail in the video for B3.3, Muscles and Motility. In neurons, calcium ions cause secretion of neurotransmitters, allowing a nerve impulse to stimulate another neuron or cell. You'll learn about this process in more detail in C2.2, Neural Signaling. Why do we have so many signaling molecules? Because structure influences function. Hormones include amines, which are amino acids with modified groups such as norepinephrine, and peptides, which are short chains of linked amino acids, such as oxytocin. It also includes proteins like insulin, which are long chains of linked amino acids, and steroids, which are lipid-based, like testosterone. Neurotransmitters can be amines, such as adrenaline or serotonin, amino acids like glutamate or GABA, or even gases like nitric oxide. A signaling molecule must have a distinctive shape and chemical property so the receptor can distinguish it from other chemicals. In addition, it must be small and soluble enough to be transported. Some signals act locally, like neurotransmitters crossing a tiny synaptic gap. Others act at a distance, like hormones that travel through the bloodstream to distant organs. For example, luteinizing hormone, or LH, is secreted by the pituitary gland next to the brain. In males, the target cells of LH are in the testes, and in females, they are in the ovaries. The effects of this hormone are very distant from its source. Signaling molecules can also be divided into two groups, according to whether they enter the target cell or not. Some signaling molecules are hydrophobic or lipid-soluble, and can easily pass through the plasma membrane. These ligands bind to intracellular receptors, which are located in the cytoplasm or nucleus. Because they are nonpolar and lipid-soluble, they diffuse across the phospholipid bilayer without assistance. Once inside, the ligand binds to the receptor and results in the formation of an active ligand-receptor complex. In most cases, this complex regulates gene expression by binding to specific sections of the DNA and either promoting or inhibiting the transcription of particular genes. Steroid hormones like testosterone and estrogen bind to intracellular receptors. Transmembrane receptors are embedded in the plasma membrane, with one part exposed outside the cell and another part extending into the cytoplasm. These receptors are specialized to detect hydrophilic ligands, like protein hormones and most neurotransmitters. Because of their size or chemical properties, such as being polar or having a charge, they cannot pass through the lipid bilayer of the cell membrane. These ligands bind to the extracellular part of the receptor, which causes a conformational change in the receptor. The inner side of the receptor becomes catalytically active and causes the production of a secondary messenger within the cell. This conveys the signal to the effectors within the cell that carry out the response. This signal transduction pathway inside the cell sometimes involves G proteins, ion channels, or second messengers like CAMP. Insulin is an example of a protein hormone which must bind to a transmembrane protein to trigger a signal response. The acetylcholine receptor is a classic example of how a neurotransmitter can trigger a rapid cellular response by causing changes to membrane potential. Acetylcholine is a neurotransmitter released by neurons at neuromuscular junctions and in parts of the brain. When it is released into the synaptic cleft, it binds to the receptors on the surface of the postsynaptic cell. These receptors are ligand-gated ion channels. This means they are normally closed but open when a ligand binds. When acetylcholine binds, the receptor undergoes a conformational change that opens the ion channel, allowing sodium ions to flow into the cell. This influx of positively charged ions causes the inside of the neuron to become less negative, reducing the membrane potential. If the change is large enough, it reaches the threshold potential, which will then trigger an action potential. An action potential is an electrical impulse that travels down the membrane. You'll learn more about this in the video C2.2 neural signaling. Another key example of a transmembrane receptor is the G-protein coupled receptors. G-protein coupled receptors, or GPCRs, are a large and diverse group of transmembrane receptors. They convey signals into cells using a second protein located in the plasma membrane called G-protein. Let's take a closer look. Here we can see the three subunits of G-protein, alpha, beta, and gamma, assemble on the receptor. A molecule of guanosine diphosphate, or GDP, is bound to the alpha subunit and keeps the G-protein in an inactive state. But when a ligand binds to the binding site on the receptor, as seen here, the receptor changes shape. This causes changes in the coupled G-protein, and GDP detaches from the alpha subunit. This allows guanosine triphosphate, or GTP, to bind in its place. Binding of GTP activates the G-protein, which separates into its subunits and dissociates from the receptor. The activated G-protein subunits cause further changes within the cell, triggering the cell's response to the signal brought by the ligand. Many different receptor functions are controlled by the G-protein coupled receptors and their associated G-proteins. The ligands that bind to these receptors are diverse and are involved in a variety of physiological processes, including sensing smell and taste, as well as regulating the heart rate and mood. Epinephrine is a key example of a ligand that binds to a G-protein coupled receptor. Epinephrine prepares the body for the classic fight or flight response. But how does it work? First, epinephrine binds to the GPCR of target cells, such as the heart or liver. This leads to activation of a G-protein on the inner surface of the membrane. The G-protein then exchanges GDP for GTP, activating it. The G-protein then moves along the membrane and activates the enzyme adenyl cyclase, and this converts ATP into cyclic AMP or CAMP. This small molecule acts as a second messenger, rapidly spreading the signal inside the cell. One example of a cellular response is liver cells breaking down glucose. This glucose is then released into the blood within seconds of receiving an epinephrine signal. The insulin receptor is another key example of a transmembrane receptor. You should be familiar with how it triggers a response. The insulin receptor, as seen here, is activated by the binding of insulin. The two tails of the protein that extend into the cytoplasm are tyrosine kinase enzymes. Kinases are enzymes that add a phosphate group from ATP to a specific molecule. In the case of tyrosine kinase, it transfers phosphate from ATP to the amino acid tyrosine in a protein. When insulin binds, it causes a structural change in the receptor and the two tails connect. Then each tail phosphorylates, or adds a phosphate to the other tail. These changes trigger a biochemical chain of events inside the cell, the signal transduction. Vesicles containing glucose transporters move to the plasma membrane and fuse with it, inserting transporters like this seen here. These transporters are channel proteins which allow glucose to enter the cell by facilitated diffusion. The glucose can then be used as a substrate in cellular respiration. A key example of an intracellular receptor that affects gene expression are steroid hormones. These hormones are lipid-based and therefore hydrophobic. This means they are soluble in lipids and are able to pass through the cell membrane. Once inside the cell, they bind to receptors in the cytoplasm, as seen here. The hormone receptor complex enters the nucleus and attaches to the DNA. This activates the production of a particular protein. For example, the androgen receptors bind to testosterone, and the resulting complex increases production of the FADS1 gene, which in turn increases production of important fats in prostate cells. Estradiol, also known as estrogen, and progesterone are also steroid hormones. They play a key role in reproduction, which you'll learn more about in the video for D3.2 reproduction. Estradiol has a broad range of effects in the ovary and the uterus, but it also acts on the brain, helping to regulate release of other reproductive hormones. Within the brain, specifically the hypothalamus, the hormone gonadotropin-releasing hormone, or GnRH, is produced and released. This hormone triggers the release of the sex hormones LH, or luteinizing hormone, and FSH, follicle-stimulating hormone, from the anterior pituitary. At different stages of the human menstrual cycle, estradiol can either inhibit or promote the release of GnRH. Just before and during ovulation, estradiol has a stimulating effect by binding to a receptor within the cytoplasm of the hypothalamus cell. Once bound, the hormone receptor complex moves to the nucleus where it acts as a transcription factor, enhancing the transcription of GnRH mRNA. Progesterone is produced by the ovary and maintains the uterine lining, which can support a developing fetus. Progesterone can diffuse directly through the plasma membrane of uterine cells and bind to a receptor in the cytoplasm. The hormone receptor complex then enters the nucleus where it interacts with DNA as a transcription factor, ultimately affecting gene expression. For example, one of these genes activated contributes to cell proliferation needed to maintain the lining of the endometrium of the uterus. Cell signaling pathways don't operate in isolation. They are tightly regulated to ensure that cells respond appropriately to internal and external changes. One of the most important ways the body controls these pathways is through feedback mechanisms. There are two main types, negative feedback and positive feedback. Negative feedback keeps conditions within a narrow range. When a system detects a change in internal conditions, it initiates a counteracting response to reverse that change, bringing the system back to a stable, optimal point, or homeostasis. In cell signaling, this often means once a desired effect is achieved, the pathway is inhibited or dialed back. This prevents overreaction and conserves energy and resources. A key example of this is the insulin signaling pathway. After you eat, blood glucose levels rise. This triggers the pancreas to release insulin. Insulin binds to tyrosine kinase receptors on target cells, like liver or muscle. This activates a signal transduction pathway that moves GLUT4 transporters to the membrane, allowing glucose uptake. As glucose enters cells, blood sugar levels fall. Reduced glucose is sensed by the pancreas, and the insulin secretion decreases. The decrease in insulin secretion is negative feedback. The signal shuts itself off once the goal, homeostasis, is achieved. Positive feedback does the opposite. It enhances or reinforces the original signal, pushing the system further and further away from the starting point. In cell signaling, this often results in a rapid and dramatic change. Positive feedback causes a self-perpetuating loop, which continues until an external factor stops it. A key example is the release of oxytocin and childbirth. Stretch receptors in the cervix, which detect pressure from the fetus, send signals to the brain. The hypothalamus stimulates the release of oxytocin from the posterior pituitary. Oxytocin then travels in the bloodstream and binds to GPCRs on uterine smooth muscle cells. This triggers a signal transduction pathway that causes muscle contraction. Contractions push the baby further down, stretching the cervix more, which causes even more oxytocin to be released. This is positive feedback. Each contraction leads to stronger signaling and more oxytocin release until the baby is born and the stimulus stops.